welcome folks, and thank you for asking me to be part of your program, Brian. So this evening, we're going to talk about the importance and considerations of formulating diets for amino acids. And so first of all, a few comments about the importance of doing this. Number one, amino acids are required nutrients. Some are typically more limiting than others, and diet can have a rather significant effect on the sequence and extent of amino acid limitation. So we're going to talk about all three of those points here. First of all, yes, amino acids are required nutrients. There's 20 of them, and we typically would classify them according to their absorptive essentiality, meaning they must be absorbed in the amounts as the animal requires them, which we would call essential amino acids, or non-essential. All 10 of those are still required for protein synthesis and other functions in the body. However, they do not have to be absorbed in the amounts as needed. The reason for that is that the body can synthesize one from another. So if there's a surplus of alanine, for example, it can be converted to glutamine or so on. And also any of the essential amino acids that are supplied in excess of need can be used to synthesize non-essential amino acids. So unless the animals are absorbing an exact optimum profile of the essential amino acids, then there will be some in surplus and they can be used to synthesize non-essential amino acids. Now, the primary function of these amino acids now is for protein synthesis. However, they have a number of other functions in the body as well. So let's talk about protein synthesis first. Now, as dairy folks, we would think about protein synthesis as the amino acids here needed now to synthesize milk protein. And that's obviously going to be a big requirement in terms of protein synthesis in the lactating dairy cow. But every minute, every hour of the day, lots of other proteins are also being synthesized, including all of the blood proteins, and I've listed a few of them there, all of the milk proteins, ion channel proteins that be present in every cell within the body, receptor proteins, messenger proteins, all the enzymes, regardless of these enzymes, have functions in protein or amino acid nutrition, but fat metabolism, carbohydrate nutrition, whatever. All enzymes are made up of proteins. Also tissue proteins. Okay, now that represents, protein synthesis represents a majority of the need for amino acids in the body. And this is where the concept of ideal balance is really important, because every time one of these thousands of different proteins in the body are synthesized, it always has the same amino acid composition. So a shortage of an amino acid in the diet or that is absorbed, then is going to have a negative impact on the synthesis of proteins. And we never quite know where that's going to be. Is it just going to be on milk proteins, or is it going to be in terms of the synthesis of some other proteins? Amino acids also involved in metabolic regulation. There's a lot of different functions of amino acids here. We'll mention a little bit of that later. And a number of the amino acids are also used to synthesize non-protein nitrogen compounds in the body, of which there's about 60 of them. And that doesn't represent a huge requirement for amino acids, but it's still a requirement. Amino acids then that are left over are going to be used for energy. They'll be catabolized, the ammonia or amino group will be released, and the carbon chains will be used for synthesis of glucose, immediate use for energy, or the synthesis of fatty acids. Now, over the years, we've identified, at least in the lactating dairy cow here, there are 3 amino acids that have surfaced as generally being most limiting for protein synthesis. That would be lysine, methionine, and histidine. Now, we really started to understand that about 50 years ago. My PhD work was published in 1976. I ran my first amino acid infusion trial in 1971. and conducted 5 experiments at that time to sequence amino acid limitation in lactating dairy cows. And our approach here was to infuse individual amino acids, combinations of amino acids into cows that were receiving just grain and forage diets, no protein supplements. So these diets were between 10.5% to 12% protein. We wanted to know what the sequence of amino acid limitation was in terms of the impact on milk protein content, milk protein synthesis, without protein supplements. And what we learned then was that lysine and methionine were the first 2 limiting amino acids, and in fact, 42% of the total response we got by infusing casein, which would represent all 20 of the amino acids, or the 10 essential amino acids. 42% of the response we got, either way there, we got just with lysine and methionine. So that gave us really clear evidence that, hey, yes, lysine and methionine are the first 2 limiting amino acids. And then if you look at the amino acid composition of proteins, you would know that once we start adding those to the diet, we're going to extend the extent of the limitation of those, because those protein supplements are typically low in lysine, methionine, or both. And we'll take a little look at that in just a minute. And then in more recent years, histidine has surfaced as a potentially limiting amino acid after lysine and methionine, especially when we're feeding lower protein diets or lower RUP diets. Okay, so let's see if we can make a little sense out of this. This table shows the essential amino acid profiles in milk, mixed rumen microbes, that'd be a combination of particle and fluid-associated bacteria and protozoa, and some common feeds. So the first line there, we have the essential amino acid profile in milk. Now let's take a look at the essential amino acid profile in mixed rumen microbes. Now, if you add up those numbers across or within a line, they're going to add up to 100%. So we're looking at each essential as a percent of total. Now, interestingly, and as I guess we would expect, right, the amino acid composition of mixed rumen microbes is pretty close to that of milk. Not exactly the same, but it's probably pretty safe to say that at least on the lactating dairy cow, on the initial start at trying to establish requirements would be to look at the amino acid composition of milk here. Now, I've highlighted in red those amino acids within mixed microbes or within feedstuffs here that are 20% or more lower than those in milk, and that's indicated in a footnote there. So I did that so it would be easy for you to identify those amino acids here in the table that are at least 20% lower than their concentrations in milk. Now, if we look at mixed rumen microbes, there's 2 amino acids that come up here. One is histidine and one is tryptophan. And as I said just a minute or 2 ago, if we're feeding lower protein diets, as they've done at Penn State and a few other places now, where they've enriched the diet with rumen-protected lysine and methionine, fed lower bypass protein diets, where mixed microbial protein is providing a majority of the metabolizable protein to the animal, histidine can come in as a 3rd limiting amino acid. So looking at that value there, that's probably not surprising. And notice that that value for histidine in mixed rumen microbes tends to be lower than in most feedstuffs. One exception there would be feather meal at the bottom of your table. So now let's just take a look at all the red values in the table. And if you scan down to each column, you're going to see more red values for lysine than any other amino acid, meaning that the RUP fraction of all of these feeds is going to have a lower, significantly lower content of lysine than in mixed rumen microbes. Okay, so it's pretty easy to see why lysine is a limiting amino acid, because RUP from these feeds will make up 30, 40, 45, hopefully not 50% of the total metabolizable protein that your cows receive. So there's a lysine issue. If you look at the values for methionine, you'll notice a lot of red values there. Isoleucine also comes up in a few feeds, like barley, like corn, like canola meal, like corn gluten meal, like cottonseed meal, and especially blood. Okay. And then you're not going to see very many other red values there in the table other than for tryptophan. And tryptophan looks like it's limiting there in mixed rumen microbes. Corn silage is really low. Other corn products is really low, as you'll see with DDGS and corn gluten meal. Okay, now tryptophan is an amino acid that we really haven't taken a close look at yet in dairy cattle nutrition, but this data would suggest that it certainly could come in as being co-limiting or limiting, certainly right after lysine and methionine. Now, other amino acids other than lysine, methionine, and histidine can become limiting in lactating cows in ways other than as building blocks for protein. And there has been a lot of work published in terms of amino acid metabolism of the mammary gland and other tissues over the last 15 to 20 years. And Dr. Mark Hannigan at Virginia Tech, he's been one of the leading researchers in this area, really looking at what is the impact of some of these other amino acids on stimulating protein synthesis, not only in the mammary gland, but some other research groups looking at that in other body tissues as well. So what these researchers have shown is that some amino acids like leucine, isoleucine, as well as methionine and histidine can have or do have potential roles in the body other than as building blocks for protein, in that they are stimulating in various different ways protein synthesis within various cells or within various tissues or organs. So many different types of in vitro experiments and using mammary tissue have been conducted to try and better understand what's going on in the lactating dairy cow. Why do some of these amino acids tend to stimulate, or what other amino acids do stimulate protein synthesis to make protein synthesis more efficient? So just in the last couple of months, Peter Yoder just completed his PhD down at Virginia Tech and conducted a really excellent study that really was to take a look to see whether or not the cows would respond in a way to a few of these amino acids in ways other than as building blocks for protein. So it was a really excellent study with 8 high-producing dairy cows, had two 4x4 Latin squares. And so 4 treatments, 4 animals, 4 experimental periods within a Latin square, and then that was replicated using a total of 8 cows. So the treatments were control, where the animals didn't get any amino acids. Lysine and methionine and histidine then were infused as one of the treatments. Isoleucine and leucine were infused as a 3rd treatment. And then the 5 amino acids were combined and infused into the animals as a 4th treatment. So amino acids were provided by jugular infusion. And the daily infusion amounts are given here. And they came up with these amounts because they did want to feed a low protein diet, as indicated in the last sentence there. It was 15.2% crude protein. And according to the latest version of CNCPS, then with that diet, the model was predicting certain degrees of deficiency of a number of amino acids, including these 5. And so they basically backfill those deficiencies then by infusing these amounts of these 5 amino acids. So note that the primary ingredients here were corn silage, alfalfa hay, corn grain, wet brewer's grain, soybean meal, citrus pulp, and soy hull. So a pretty simple diet, but yet not an uncommon diet, at least to us here in North America. In terms of production results, and this is the only slide that I got here on the subject, or to show the results of, but which is milk, effects on milk protein content, and milk protein yield responses. So as we would expect, these cows were beyond peak lactation, and you provide those animals with additional lysine, methionine, and histidine, we would expect to see some sort of a bump in milk protein. In fact, there was a significant increase in milk protein, as you'll see in the second bar graph there. No effect on milk yield, that's not surprising. We often would not see an impact on milk yield. But because of that significant increase in milk protein content, there was a significant increase in milk protein yield. Now notice that with isoleucine and leucine had no effect on milk protein content. So as we've been kind of looking at things over the years, we would have said, well, isoleucine and leucine were not limiting amino acids. Okay, there was no impact on milk protein content. However, there was a significant increase on milk yield. So you multiply those two values together and you get a significant increase in milk protein yield with the 5 amino acids. Now, what's really exciting here is that when you combine those 5 amino acids together, now you get the milk protein content response back, you retain the increase in milk yield, and now there's an additive effect in terms of the impact on milk protein yield. So here we have 2 groups of amino acids that are independently and additively increasing milk protein yield. And as the authors have appropriately stated, this then creates doubt about what we have been thinking as the single limiting amino acid theory. In other words, amino acids can be co-limiting, but in different ways, having different impacts on the mammary gland. Secondly, the importance of formulating diets for amino acids is that achieving a more optimum balance of absorbed amino acids reduces the risk of a singular or a small group of amino acids being more limiting than other amino acids or other nutrients, as well as increasing the efficiency of use of absorbed amino acids for protein synthesis and other metabolic functions. So here we have essential nutrients. We know it's important to formulate for them, to provide the animal a good balance. And by providing a good balance, we're doing 2 things, okay? We're minimizing the possibility that those most limiting amino acids are going to be limiting in the diet. And we're allowing the animal an opportunity to increase efficiency of use of those absorbed amino acids. So the result is going to be an improved animal performance and a reduced need for supplemental RUP. Now, we understood the importance of amino acid balancing from the swine and poultry literature back in the 60s and early 70s. So Dr. Rulkin in France, way back in the later 70s, 80s, began to infuse lysine or methionine into the abomasum or into the jugular vein of lactating dairy cows. And his goal was to establish what is the ideal profile or amount of lysine in digestible protein or ideal amount of digestible methionine. So then in later years, as they designed a model that would predict amino acid flows to the small intestine and could predict the concentrations of lysine or methionine or other amino acids in digestible protein, they took that information and run all of those diets through their model. And so what we see here is a residual yield response plot. And mathematically, it was determined that the ideal amount here of lysine in digestible protein was 7.3% of that digestible protein. And so they took their other set of studies, and it wasn't necessarily that they conducted all the studies, but they certainly did conduct a majority of those studies. And here they looked at infusing incremental amounts of methionine across a number of different studies. Note in the footnote, 265 cows were involved in this data set, and the optimal amount of methionine then in the digestible protein was 2.5%. So just remember that 7.3, go back in 2.5. And some years later, we at the University of New Hampshire ran a number of these studies ourselves. Some other people here in the United States did. And so as a member of the NRC committee that was charged with introducing an amino acid model then, within NRC, I needed to do something similar to this. So we took the data that we had and we went through this same set of, or we used the same procedure to establish these residual plots of being able to combine the data, and we came up with an optimum value of lysine of 7.2, very close to what Dr. Rulkin had come up with in terms of the European or the French model, using a different data set and using a different model for predicting content of lysine in NP. And at this point, we did this with a beta version of the NRC model. And we took our methionine studies and we came up with this plot. So 7.2 and 2.4, values very similar to what Dr. Rulkin had come up with. Then after the NRC came out and there was no more changes being made to the model, we redid all of our analysis here, and notice our values for lysine and methionine dropped a little bit from 7.2 down to 6.8, 2.4 down to 2.29, but still very close to a 3-to-1 ratio. And then there were some revisions made to the model a few years later. That really didn't change things a whole lot. And then several years later, and we shared that at ADSA meetings in 2009, we took the very same studies, and evaluated the diets and everything with CPM dairy. So we could arrive at these optimum values for CPM dairy. Notice that the values for both lysine and methionine were higher than NRC. So that represents model differences, folks. That does not represent any differences in the data set that was used. And then as the more recent versions of CNCPS came available, we did the same thing with those models, and you'll see the values here. So anyways, those are the target values that we've come up with using the same data set. Now, what's the benefits then of using that information? What's the benefit if we try and optimize lysine and methionine nutrition using those values that I just gave you? Well, what we've learned is that we definitely can reduce the risk of a deficiency of the most limiting amino acids, which would be lysine and methionine here. The average sort of responses in early lactation cows in terms of milk yield would be about 2 to 4 kilograms more milk. Some studies have shown more than that, a number of studies less than that. But for early lactation cows, where we start supplementing these amino acids at the time of calving or before calving, that's what we can pretty much expect to see. Obviously, that's going to depend on what the diets were, or the diets that were fed prior to the initiation of amino acid balancing. Increased milk components, especially milk protein. 0.1 to 0.2 percentage unit increase. Sometimes we see 0.3. We see 0.3 increases both in research and in several dairy farms across the U.S. And 60–70% of the time, an increase in butterfat. And lastly, healthier and more productive transition cows. And if you watch the presentation by Dr. Osorio, he was sharing data where they fed diets that tended to have higher concentrations of lysine in metabolizable protein, and then supplemented those transition cows then with a supplemental methionine. And if we were to summarize not only all the data that Dr. Osorio presented, but other data as well, this is what happens when supplemental methionine is provided to these transition cows. So these cows would have been methionine deficient. They were receiving diets that had higher levels of lysine and metabolizable protein, not as high as most of us would like to have seen. So we really feel that some of these benefits of methionine supplementation could have been greater than what they were. But nevertheless, these are most of the benefits, all the way from increased feed intake to having an impact on health, immune function, inflammation, reduced oxidative stress. Larger embryos, decreased pregnancy loss in a few studies. There's more studies going on in this area. And What's really exciting, I think, is the impact here on the offspring. Offspring born of mothers in which supplemental methionine was provided to the dams or to the mothers. Okay, so that gives us some evidence here that improved methionine nutrition, and then probably as we look deeper in the future on improved lysine nutrition, histidine nutrition, we might see some of the same things here, the impact it can have then on fetal programming. There was some older data that existed. Most of these studies would have been conducted in the earlier 1990s as Ajinomoto was evaluating one of their early versions of a protected lysine supplement. So they were using that in line or along with Smartamine M just to see if they added some lysine and methionine to early fresh diets, what would be the impact on lactational performance. And across those early studies, the average yield in milk protein was 3.8 kilograms. Now, lysine has, I think, really become increasingly more important in dairy cow nutrition. We're feeding more corn silage and less legume and grass forages, certainly here in the US over the last 10, 15 years. Corn silage is low in lysine. We often replace soybean meal products with canola meal because of price. Well, canola meal tends to have a little lower concentration of lysine than what soybean meal does. Blood meal has fell out of favor for some producers, which has a really high content of lysine. Often we like to feed distillers grains and other high protein corn products in place of other protein supplements because they're less expensive. Okay, they're low in lysine. And we're probably getting a little more aggressive in recent years with feeding rumen-protected methionine supplements. So we may very well be hitting the ceiling quite quickly in terms of not having an adequate supply of lysine to really make use of the RP methionine supplements that we have available to us. We've also got a greater desire to feed less RUP than before. We're also learning that lysine may not be as available in our feedstuffs as we thought. In other words, maybe some of our feedstuffs has a lower content of lysine than we really thought they had, and that could very well be true for something like our corn that we feed our animals and soybean products. I was just talking with Dr. Hans Stein, University of Illinois, a couple of days ago, and he said a trend that they've seen is reduced lysine and amino acid concentrations in both corn and soybeans. And he thinks it could very well be a result of the increased yields that farmers are getting in terms of bushels of soybeans per acre, bushels of corn per acre, lowering the amino acid content of those feeds. And we know that milk and milk protein yield is very closely tied to absorbed lysine supplies. Lysine basically only has one function in the body, and that's protein synthesis. It does have some other functions, but they represent a smaller part of the total need for lysine. So just remember, lysine, primary function is for protein synthesis. And just to kind of drive home this point, back years ago, when I was at UNH, way before I ever left UNH, we had a few students that wanted to do a senior project, or they started a master's program and wanted to do an initial study. And so they took a look at supplementing diets with Smartamine ML. That was the original Smartamine ML that Adiseo had. Put some Smartamine M in the diet as well to make sure that Smartamine M was, or that supplemental methionine was where we thought it needed to be. And so that really created a somewhat greater deficiency of lysine. But these were, well, these cows went on trial at on average 106 days in milk. And notice the impact that that supplemental lysine had on milk yield, really pushed milk yield, as well as increasing milk protein content. The next experiment was a smaller experiment, only involving 5 cows, but nevertheless, we saw the same effect. We set the study up the same way, adequate methionine, at least what we thought was adequate methionine at the time, and then brought in incremental levels of supplemental lysine.
And in this trial here, we did not use smartamine ML, but here we did use infused methionine to create those concentrations, those increased concentrations or supplies of lysine to the animal. But the diets did have smartamine M in them. So just making a point here that one of the reasons here for amino acid balancing now is to improve lysine and methionine status in particular. We'll be focusing on other amino acids in the future, I'm sure. But another reason for doing it is to increase efficiency of use of metabolizable protein. And here I've gone through an exercise to show you that there's different ways of achieving 180 grams of metabolizable lysine and 60 grams of metabolizable methionine. So if you happen to be using the NRC model and your cows are given roughly 90 pounds of milk with 3132% milk true protein test, that's about how many grams of lysine and methionine those animals are going to need. Now, you can achieve those 180 grams of lysine and 60 grams of methionine several different ways. You can feed a high corn DDGS diet, which would be the first diet in your table. Low content of predicted lysine and metabolizable protein, fairly high content of lysine and metabolizable protein at 1.9, no amino acid supplementation there. That is a high corn diet. Now, we know that a diet like that works out in the field. However, we also know that you have to feed more RUP. The dietary crude protein content of that diet is going to have to be higher. And if you go through the math, as I've indicated here in the footnote, notice that that diet, according to these calculations, require about 8.1% of dry matter as RUP. Now, you add that to an RDP requirement of about 10% of diet dry matter, and you're up to an 18% protein diet. Or you can go the other way and say, you know, I really want to push lysine and methionine. I want to optimize my diets. I want to improve efficiency of use of metabolizable protein. So I'm going to shoot for 6.9% lysine and MP and 2.3% methionine in MP. Notice that the MP requirement now fell from 3,157 grams in the second column all the way down to 2,609 grams. That is a huge saving on metabolizable protein. And that's all, that saving is going to be on RUP. You don't want to do anything to negatively impact the animal's ability to provide metabolizable protein to herself from mixed microbial protein. So now if you go all the way over to the right-hand column, you go through the calculations there, that allows lowering RUP in the diet to 5.4%. So what do we have? We have a diet here of 5.4%. RUP, RDP is going to be around 10, maybe a little bit higher than 10, depending on what works best for you. And you're at a 15.5% protein diet. So a tremendous opportunity here to save feed costs on the purchase of supplemental RUP, and we've done it through improved efficiency of use of MP. Now, we knew this to be true. Years ago, after the NRC came out, we put a number of diets through the model, and we just looked at the difference between actual milk in these cows and MP allowable milk. And interestingly, as the model predicted increasing amounts of lysine and metabolizable protein, so we picked studies here that had different types and amounts of protein supplementation. Notice that if lysine and MP was really low, on average, cows gave a little less milk than what the model says was MP allowable. But as we increased lysine in metabolizable protein, now the cows began to give more milk than what the model said via MP allowable milk. Okay, and the same thing with methionine. So when methionine was low, cows tended to give less milk than what the model said the cows ought to give via MP allowable milk.
But as the amount of predicted methionine and MP increased, the cows gave more milk than MP allowable. And I'm just going to show you one study out of many examples from the literature of reducing dietary RUP by amino acid balancing. And this work was out of the University of Wisconsin. They fed as their control diet what they call the traditional protein diet: corn silage, alfalfa silage, high moisture corn, soybean meal, distillers grains, some expeller soybean meal. Okay, so some complementary proteins in there. Diet was 16.9% protein. NRC model predicted that diet would provide 160 grams of MP lysine. Then they formulated a lower protein diet. They took the distillers out, expeller soybean out, increased solvent extracted soybean meal, put in a little high moisture corn, lowered protein down to 15.7%. Notice that the model, though, however, was calculating the same number of grams of MP lysine, 161 grams. But the predicted flows of MP methionine fell a little bit, and the lysine to methionine ratio increased. So the whole goal was here now was to backfill that deficiency of methionine with some supplemental methionine sources. In this case, they used Metasmart or Smartamine M. Well, they used both as two separate treatments, and increased the methionine supply from the original 48, or what was in the low protein diet, 45, up to 54. So from an amino acid standpoint, we have an improved diet, don't we? We did not lower model predictive flows of MP lysine, but the cow is getting more methionine via an increased lysine to methionine ratio. And if we go right to the results here, and I'm just going to show you a few of the results, but just go to the bottom of this slide and notice MP balance. So with the traditional protein diet, that was designed to basically have an MP balance of 0. It's very close to that. But then when they pulled out some of the protein, those animals then became negative in terms of NP. So the model was saying these cows are going to give less milk because they had a negative NP balance. Well, the model doesn't correct for NP requirements based on amino acid balancing. Okay, so now if you look at milk protein yields, or if you look at milk yield, there was no differences amongst those treatments. Milk protein content was actually higher with the last two experimental treatments there with Metasmart and Smartamine M. In terms of yield of protein, numerically, those values were higher for those two treatments as compared to either the traditional protein diet or a low protein diet. Milk nitrogen to feed nitrogen ratios improved. So the bottom line here is the animals on the lower protein amino acid balanced diet here did just as well, if not better, than with the traditional protein diet, even though the model would say that those cows would not do as well. So there's some take-home messages here. Reduced RUP feeding saves costs for purchased protein supplements. Reduced RUP feeding provides an opportunity to feed more of the rest of the diet, which should be expected to increase microbial protein synthesis. And really, if you think about it, amino acid balancing is the only option we have for selectively increasing efficiency of use of RUP. So you have to decide as a dairy producer, as a nutritionist, do you want to try and improve efficiency of use of RUP or not? Is it to your benefit? And the last point here, just to remember, is most ration formulation models do not adjust model predicted requirements for MP as a result of amino acid balancing. Which means you cannot trust your model now on predicting milk yield based on supply of metabolizable protein. And if you're using a good model, you should be able to consistently run negative MP balances as a result of amino acid balancing. So how can we be successful here? And this is really where considerations come into play. Number one, you've got to let go of balancing diets for crude protein and instead balancing for rumen degradable, feed protein, RDP, and the most limiting amino acid.
as a dairy producer, as a nutritionist, you should know in every herd, what is the RDP content of your diets? Is it 10%? 9.5% of diet dry matter with the model that you're using? Is it 11, 11.5? But that's a value that you should always be watching. Okay, because that's a requirement for rumen microorganisms. And the cow now requires amino acids. RDP is for rumen microbes. Amino acids, and in terms of metabolizable protein and the amino acids contained within that, that's what the cow needs. Now, we've known for a long time that the relationship between the crude protein in milk, or milk crude protein yield, and crude protein intake, that relationship is very, very poor. Now, we've known that ever since I started graduate school in 1969. That was a very poor relationship. And then over the years, as we introduced the metabolizable protein concept, you can see here now that the relationship between milk protein yield and model predicted MP supply is somewhat better than with crude protein. So some improvement there. Now, if you go to the bottom of this slide, you can see the relationship between actual protein yield and model predicted flows of methionine or model predicted flows of lysine. Notice how much tighter, excuse me, those relationships are than with metabolizable protein. Also notice that the relationship here between milk protein yield and model predicted flows of lysine is even stronger, better than it is with methionine. That goes back to the point that I made. Lysine has one primary function in the body, and that's as a building block for protein. The animal doesn't have enough, she cannot make that milk protein. You supply her more, and that's a limiting nutrient, she will produce more. And so we knew that, and because the NRC model did not predict requirements in terms of grams per day, after the NRC was released, we ran a lot of studies, a lot of diets through the NRC model initially. We ran 300-400 diets through with different production levels, different amounts of protein in the diet, because we wanted to look at the relationships here between actual milk and milk protein yields relative to model predicted flows of MP, MP methionine, MP lysine. And then some years later, we added another 400, 500, 600 diets, and we improved our relationship. But I don't have time to get into that, other than to say, just like you saw in the previous graph, protein yields were more accurately related or predicted from predicted supplies of MP methionine and MP lysine than from metabolizable protein. And again, this all makes sense because what the cow requires are the individual amino acids. The cow does not have like an absolute requirement for metabolizable protein. Okay, it's the individual amino acids that she needs. Achieving success also requires us to understand that the digestibility of RUP is important, or that the digestibility of amino acids in the RUP fraction of feedstuffs is important, and that is not 100%. And Dr. Mike Van Amberg talked about that. He made a big deal in his presentation, and he actually showed you a study where they depressed RUP digestibility of blood meal and showed us the impact that doing that had on animal performance. So RUP digestibility here is important. And we need to model our protein supplements and our feedstuffs, make sure that we get our RUP digestibilities as close to what they actually are as possible. Okay, but we also need to understand that there's differences in the amino acid digestibility, or there's differences in the digestibility of the individual amino acids that are in feedstuffs. And just very quickly, I want to show you some data that indicates that. And I got this data and I'm giving you this information. This is Dr. Hans Stein's website at the University of Illinois. And if you go to his website, you'll see that he's got an feed ingredient database. So if you click on feed ingredient database, you're going to find Lots of spreadsheets there, lots of spreadsheets showing the nutrient composition of different feedstuffs as determined in swine. But the reason that I'd like to have you go to that website and just take a look at what they have reported there on ileal digestibility values of selected feedstuffs as determined in pigs, and they have the same set of data for poultry. Now, why am I showing you this? Well, if we look at dried brewer's grains here, notice the digestibilities of the individual amino acids are all pretty close together. I think I see a high there of 92% or 95% for arginine and a low of 82% for lysine. Now, notice that lysine tends to be a little bit lower on average. The average here for dried brewer's grains is 87%. Notice lysine is 82%. Go to color or corn, yellow dent corn. Now, on average, digestibility as determined in pigs is 82% across all 10 of those amino acids, but notice lysine is the lowest. The values that I've highlighted in red here are at least 10 percentage units or 10% lower than the average. So that 74% is 10% or more lower than the 82, okay? So I did that just so you can see very quickly here, just perusing the table, which amino acids tend to have the lower digestibility values. Corn, distiller's grains, lysine is also low again, 76 instead of 81, but the amino acid having the lowest value is tryptophan. Okay, so you can just look at that table. The point is here is that there are differences in amino acid digestibilities across feeds, and there's differences within feeds for the 10 essential amino acids. This table shows a comparison of digestibility values for canola meal, rapeseed, and some soybean meal. Soybean meal, we've recognized over the years, tend to have very high digestibilities. You'll see the average digestibility values there for those 2 soybean meal samples. It's 87 and 86%. And pretty uniform in terms of digestibility across those 10 amino acids. However, if you look at canola meal, you'll see the average digestibility value is a little bit lower there than for the soybean products, and that would include rapeseed meal as well. Okay, threonine tends to be pretty low in that one canola meal sample, the one on the left. But that's really the only amino acid that stands out here in this table as being what I would say significantly lower than average. And here's a comparison of some digestibility values of animal protein supplements. So blood cells, spray dried blood cells, blood meal, feather meal, hydrolyzed feather meal with blood or without blood, and then a really high quality fish meal. High quality fish meal, as we would expect, would have very high digestibilities. We wouldn't expect to see much of a difference in digestibilities amongst amino acids. But excuse me. The blood cells are spray dried. Okay, heat was applied. Notice a very low digestibility value there for isoleucine. The next blood meal sample also lower for isoleucine than the other amino acids. A feather meal tended to be the digestibility value for histidine and lysine on average were lower. So if you go across all these, lysine kind of comes out as that one amino acid that tends to have a lower digestibility value as compared to the average of the other amino acids. I'm emphasizing this in part because we know that heat can have a negative effect on protein digestibility and amino acid availability, and a lot of the feeds that we feed in the ruminant industry, including dairy cattle, have gone through some sort of a drying process. Here in the upper Midwest, last year, almost all of the corn that was harvested for dry corn would have gone through a dryer. What effect does that have possibly on lysine digestibility? We really don't know. We do know that there are 3 different types of reactions that occur in feed as a result of heat processing. The protein cross-linking reactions would be the type of reaction that we would see in something like a blood meal product, for example, that is primarily just protein, doesn't have a lot of carbohydrate or any carbohydrate in it. So any of the depressed digestibility there would be as a result of protein cross-linking reactions. And the Maillard reaction as being the most important reaction that would occur in other feedstuffs. So lysine is the most vulnerable here because it has that reactive epsilon amino group. And that's the group then that's going to get tied up either with sugars, reducing sugars in the case of the Maillard reaction, or in the protein cross-links in higher protein containing feeds. And just very quickly, Sarah Boucher, when she was at the University of New Hampshire for her PhD work, she used the Rooster assay out of the University of Illinois and actually did that work down at the University of Illinois with Dr. Hans Stein down there. And she wanted to take a look at the impact of heat treatment of two different sources, a solvent-extracted soybean meal, expeller soybean meal, and then 4 sources of distiller's grain. So those are the feeds that she had collected. And then she took one sample of each of those and exposed them to some additional heat, the conditions of which are described here in the slide. Okay. And the reason for doing that was not only to look at the impact of heat treatment on depressing digestibility in the RUP fraction of feedstuffs here, but when RUP digestibility is depressed, does it have a greater effect on lysine than on the rest of the amino acids? And that's exactly what this slide shows. Okay, if you go to the right, look at total amino acids, the first 4 bar graphs there, that would be the protein supplements without any additional heat applied. Then you apply some heat, you get depressed digestibility in that 1 soybean meal sample, but notice if you go to the left, lysine digestibility was depressed more than RUP digestibility. Okay, and then she took the other sample there, applied heat, got a depressed amino acid digestibility value, but lysine digestibility was depressed more. And she also looked at 4 different samples of DDGS, and she took one of those samples and applied additional heat and noticed a really pretty serious negative impact on lysine digestibility. One of those DDGS samples already had a depressed lysine digestibility, as you can see. It was not necessarily picked up if you just looked at total amino acid digestibility, but if you looked at lysine in particular, yes, it was depressed. I'm making this point because we tend to feed protein supplements that have been heat processed, and there's always that possibility here that lysine digestibility is less than the other amino acids, or for the other amino acids in that RUP. So just be mindful of that. And this is a figure that I'm sure that most of you have seen, some work by Dr. Norman St. Pierre years ago, where they looked at the influence of depressed RUP digestibility in blood meal and the impact it had on RP lysine digestibility. So if RUP digestibility of the blood meal is really, really high, then the digestibility of lysine is high. But as RUP digestibility goes down in blood meal samples, notice that lysine digestibility continues to be depressed even more. So if you focus at an RUP digestibility of 40% there, the lysine digestibility on average was 20. Okay, so these are just things that you want to keep in mind if you want to have success in terms of amino acid formulation. Always be thinking about what is the lysine availability. Are the lysine values for my feeds in the feed library, is that correct? Does that represent the lysine levels that I have in my feedstuffs? Number 3, in terms of amino acid balancing, really important here to use reliable estimates for the bioavailability, if you will, of amino acids in our rumen-protected amino acid supplements. Very important. Because if we're going to be successful here with amino acid balancing, we're obviously going to be employing the use of rumen-protected lysine and methionine supplements. And so do you really know what the bioavailability of lysine or methionine is in the products that you're feeding? Is it what the supplier tells you that it is? Or is it something less than that? And we started getting concerned about this back over 20 years ago at the University of New Hampshire. We really felt that this whole concept of amino acid balancing and lactating dairy cows was going to progress. And we'd probably always be behind that of swine or poultry, but nevertheless, there would come a time when people would be interested in feeding less bypass protein, lower protein diets, and so amino acid supplementation is going to get more and more important. So we kind of figured that more and more products would be coming out on the market. What methods are available to evaluate these products? Well, I've listed a number of the methods here, and I've listed the shortcomings, and we considered these different methods. But nevertheless, they're in vitro or they're in situ methods, with the exception of the last three. The plasma-free amino acid area under the curve. Well, this is where you put into the rumen a pulse dose, maybe 50 grams of methionine or 100 or 120 grams of lysine. And then you take a lot of blood samples and you look for the spike in lysine or the spike in methionine in the blood plasma. And then you calculate the area under the curve. So you're doing that with either infused lysine or methionine or by providing a product. And so the area under the curve then, that gives you an idea of how much of the lysine or methionine was being absorbed, okay? But you're relying on the blood, so it is an animal bioassay. However, that's not the way we feed these products. And so we really didn't feel that this was a good method going forward for the future. We could use something like the milk protein dose response method, but the problem there is we have to make sure that we maintain amino acid deficiencies in all cows over the entire range of the dosages used, and that's always impossible to confirm. So what biomarker can we use in which increased absorption of an amino acid will reflect a change? Increased amino acid absorption may not reflect a change in milk protein if the cows are not deficient in that particular amino acid. However, we learned early on that those animals will respond with an increase in either lysine or methionine or whatever we're providing those animals. So what's the shortcoming of that approach? Well, really not a whole lot, other than the fact it takes rumen cannulated cows. And as long as we can demonstrate beforehand that the responses between increasing amounts of absorbed lysine, methionine, histidine, or whatever results in a linear increase in blood plasma concentrations, then we've got ourselves a biomarker. So we really felt that a robust animal-based method was needed. We wanted a system where we can put the products right into the diet. We can mix it in with the diet, whatever, because that's an important part of the whole evaluation process. And we know it's just difficult. And we realized way back then it's very difficult to protect these amino acids from ruminal degradation and yet have them be available for intestinal absorption. Not just available, but be digested and be absorbed. So the decision which product that you folks buy should be based on knowing not only the cost of the product and not really what the supplier may or may not be saying is the bioavailability, but what is the actual bioavailability value as determined under experimental conditions using a standardized protocol. And the method needs to be one that you can use for liquid products like Element, for example, or liquid Metasmart. Now, sort of a surprise, I think, to the industry, because nobody had really done this kind of a study before. And this was work out of Minor Institute, where they looked at the effect of on-farm feeding practices on the bioavailability of lysine and rumen-protected lysine products. And I'm just going to show you the results here of the first study.
And what they did in this experiment, and they had actually 2 experiments in that particular study, but in this experiment, their goal was to look at, well, how much lysine is released from 6 different rumen-protected lysine products when incorporated into total mixed rations that differed slightly in dry matter content, and those dry matter contents are indicated on the right-hand side of your slide. Okay, so what we're looking at here is the loss of lysine in the total mixed ration. Okay, this is not lost in the rumen. This is lost in the total mixed ration. How much of the lysine in those products were they able to recover in the TMR after those products were exposed to the TMR for 6, 18, or 24 hours? And these results were pretty shocking to some people. But the point is, it really demonstrates here something, and that is we have to consider in evaluation of these products the exposure of these products to the diets that we're feeding our animals. If you feed one group TMRs and you're putting your amino acids in those one group TMRs, then you need to have some understanding of what is the stability of those amino acids in that TMR all the way out until the cows are fed again, which would be 24 hours later. So this is very important data. So with all of that behind us, then I'm just going to explain very quickly the dose response method that we use at the University of New Hampshire. And so we use high producing dairy cows, and Nancy Whitehouse at the University of New Hampshire, she's been involved in all of these trials and over the last 20 plus years. So we've adopted a very standardized protocol. What's important here is that these supplements are put into the TMR before feeding. So they're exposed to the TMR 8 hours before feeding. Cows are milked at 8-hour intervals. They're milked at 12-hour intervals. I mean, they're fed at 8-hour intervals. And then blood samples are collected the last 3 days of each period. So very standardized protocol. So the experiments are set up in such a way that we've reached steady state. The animals are consuming these products 3 different times over the course of a day. The products are being exposed to the TMR. Here's the results of one study where we evaluated the first generation product AgiPro-L. So in this particular study, we had 6 treatments. Animals, and this would have been a 6x6 Latin square, okay? And so one treatment, the animals didn't get any lysine. One treatment, animals received 30 grams per day of infused lysine. Third treatment, 60 grams per day of infused lysine. That's continuous infusion over the course of a 24-hour period for 7 consecutive days. And the fourth treatment, animals received 30 grams of lysine from AgiPro-L, 45 grams, and 60 grams. So 3 different levels of AgiPro were fed there. Notice linear increases in plasma lysine for both feeding the product as well as for our infusion treatments. So if you take the slope of the dose response line there for the AgiPro treatments, that slope is 0.0068. Divide that by the slope for infused lysine, and you get a bioavailability of 37%. So this is the approach, okay? Use the same approach for every amino acid that Nancy has evaluated. So this is an evaluation of the 2G and the 3rd generation product. And infusions were also part of the treatments. Here we evaluated SmartAmine ML. This was the old SmartAmine ML that Adiseo had, so we wanted to evaluate the lysine in that up against infusion and another supplement. And you see the bioavailability is calculated at the bottom of your slide. This shows the bioavailability of lysine in the new SmartAmine ML product, and got a bioavailability of 80.8%. So Nancy summarized for me all of the studies
and the average bioavailabilities of all of the lysine studies. So what you see here, the number of products that were evaluated, or the number of tests made. In some cases, one product might have been evaluated more than once. And on the x-axis, we got the calculator, or the predicted bioavailability values using this technique. So notice that the bioavailability values for most of the products is pretty low, not surprising. The two values there of 80 and 85% is the bioavailability of lysine in the new SmartAmine ML product. Same approach used to evaluate rumen-protected methionine products. And this shows an evaluation of SmartAmine M and bioavailability here of 82.8%. Notice the very low standard error bars. So as Nancy has gotten better and better in terms of refining everything that she does, more and more precise estimates of bioavailability. Here, Nancy evaluated SmartAmine M coming out of their older plant, their new plant, no differences up against MEPRON and AminoSure M. And a more recent trial here, doing a comparison of bioavailabilities between SmartAmine M, AminoSure M, XM, I should say, MediPearl, and Timid. And a comparison of bioavailabilities of methionine, SmartAmine M, and ML. Again, getting bioavailability values of 80 or slightly higher in those two added sale products. The technique can also be used to evaluate the analog products. First thing we had to do was demonstrate that we got linear increases in total sulfur amino acids plus the analog itself as measured in the plasma as we infused increasing amounts of the analog. Okay, and here's a first stab at the evaluation of MetaSmart bioavailability here in this trial of 50.3%. And here is a summary of the studies, the methionine studies that Nancy has done. So at the end of the day, why are we doing this? Well, let's use methionine products as an example. So currently in the US, we have a number of different methionine products. Okay, we know what their methionine content is. We know that. The question is, what's the bioavailability values? Do you want to see independent determinations of those bioavailability values? Well, if you go back some slides, you'll find values for most of these products, and you can put them into this table. Then you can take column 1 and column 2 and multiply them together, and that'll give you the metabolizable methionine content within that product. Okay? And then from that, you can determine what is the relative feeding rate relative to SmartAmine M. I don't think anybody would disagree. SmartAmine M has the highest bioavailability value of any of the protected methionine supplements. So we'll give that a 1x. So do you have to feed 2 times more, 3 times more, let's say, of MEPRON to deliver the same amount of methionine? Well, the data is here. So you can go through those calculations and see what the relative feeding rates are. So if you know the relative feeding rates and you know the cost of the product, you can make some informed decisions here about which product you wish to feed. So take-home messages here, don't feed any of these products. We would suggest you don't feed any of these products unless you've seen confirmed estimates of bioavailability using the amino acid dose response method as described. Make sure that when you see those results, that there's confirmation that those amino acid supplements was mixed and consumed with the rest of the diet during the study. Do not accept comparative milk production data as proof of claims of amino acid bioavailability. And suppliers really like to show you that data, show that both products, their product and a superior product, resulted in the same level of milk protein, and so therefore the product is just as good. However, such data always favors inferior products. Why is that? Well,
because cows respond to increased absorption of an amino acid only if that amino acid is the most limiting factor for production. Otherwise, the cows won't respond. Okay? Therefore, if you're going to use the production data and a comparison of different sources of that amino acid, all cows fed the best product must remain deficient in that amino acid after supplementation, and no other factor, be it heat stress, some other dietary factor, can be limiting production. Well, that's absolutely impossible to achieve. So if that isn't the case, and you can't do that with any degree of certainty, then the superiority of the best product won't be seen, and the inferior product relative to the superior product will look better than they are. And I'm just going to show you one example here of 4 products, okay? So let's look at milk protein. We got our negative control, and then we're evaluating 4 different supplements, looking at effects on milk protein. Well, product A has a value of 3.44% milk true protein, product C 3.46. You would say those 2 products are not different, right? Milk protein concentrations are the same, 3.44, 3.46. However, if you go to the bottom of the table, take a look at plasma methionine concentrations and plasma total sulfur amino acid concentrations. Product A, the value is 102.5. Yes, higher than the 95.4, but that's an increase of only about 7 units of total sulfur amino acids in the blood. Now go to product C. Notice the value there is 122.6. That is significantly higher than the 102.5 value. And if you just go through the math here, you would say that the bioavailability of product A is probably somewhere around 25 to 30% that of product C. Okay? So if you look at the milk performance data, in this case milk protein content, you say those products are the same when in fact they're not the same. Achieving success also means using correct model inputs, and Dr. Mike Van Amberg, he explained that. It's really important to make sure that all of your production and animal inputs are correct. Also make sure that all of your feed inputs are correct. If you want your model to be as close as possible to being right, you got to make sure that all of your inputs are correct. And lastly, it's really important as we get more and more into amino acid balancing to use ration formulation programs with optimization capability, especially those that will give you model compliant solutions. So this is kind of a big deal. You still want to make use of your guidelines for a targeted profile of absorbed amino acids in MP, as well as for targeted gram supplies relative to some model predicted supply of those amino acids up against available energy. And Dr. Mike Van Amberg talked about that as well, and you see that in the right-hand part. So you want to keep these values in mind. So just a few quick comments, and then we will wrap up here. Comments regarding reformulation of diets through optimization. Too many people think amino acid balancing is too costly, and therefore that's why they don't want to do it. Well, the reality is it doesn't have to be that way. We now know that implementing amino acid balancing by using rumen-protected methionine supplements in combination with higher lysine containing protein supplements, or using a combination of rumen-protected lysine and methionine supplements together, does not always have to significantly increase diet cost. It really depends on what you're already feeding. In fact, you may be paying for the cost of amino acid balancing, but you're not amino acid balancing, and you're not going to know unless you look at an optimized reformulated diet. So the approach here would be know exactly what your current diet costs are. Know that. Then reformulate that diet through optimization to get a least cost or optimized reformulated diet that supplies the same amount of the most limiting amino acid, which is usually going to be methionine. Now, when you go through that process, make sure that you consider all the available feeds and supplements. Maybe you'll find that as a result of optimization that, hey, the model is pulling in some feeds in a little higher amounts or maybe a little less of this or that.
than what you thought was really optimal, and you're able to save some money. Maybe you're feeding a surplus of something, and now you realize, maybe I really didn't need that much RDP in the diet. I can take a little bit of that out and save a little bit of money. Okay, now we would typically call that the cost window diet. Now that diet now should be less money than the diet that you originally started with, and it should be just as good, if not better. Now the difference in cost between those two diets is the money you have to spend for amino acid supplementation and amino acid balancing. So that's the approach that we've used here in recent years. It really all depends on the diet that you're already feeding, if you're not already amino acid balancing. In some cases, that cost window diet has saved producers, or that cost window diet is actually less costly by 40, 50, we've seen as much as 75 cents a cow a day. That gives you a tremendous opportunity then to go forward with amino acid balancing. But typically, that cost window diet is going to be closer to 25, 30 cents less, but sometimes higher than that. And lastly, use cow feedback to fine-tune your diets. In other words, correct for model prediction errors. No models that you folks are using or that we use are perfect. So regardless of how good we think the model is, we still need to rely on cows to give us some feedback. And there's a number of things, of course, we can watch for. We look at the influences on dry matter intake, milk yield, whatever we're looking at in terms of rumen function, and you see the list here. But the questions you need to be asking yourself after you're feeding your improved diets, are you really feeding enough RDP? Are you sure? Are you sure your model is right? You definitely want to make sure that you are feeding enough, and you won't know unless you challenge the cows with a little more RDP. That's why it's so important in terms of tracking the amount of RDP in your diet. Are you feeding enough supplemental methionine? You're achieving the right lysine to methionine ratio, but are you sure the cows won't respond to a little bit more? You won't know unless you challenge the cows. Okay, and you got milk protein concentrations to look at. Will your cows respond to additional lysine? Well, I think as the years have gone on, I think there's a lot of cows out there in the world today that would respond to additional supplemental lysine, but we haven't challenged those cows. We haven't challenged them. We're just looking at the lysine to methionine ratios. And maybe we're not even at the optimum model predicted levels of lysine and metabolizable protein. So if you're not already there, challenge those cows. What are you going to be looking for there? An increase in milk yield, increase in milk protein yield, possibly an increase in milk protein concentrations. Are you feeding more RUP than you really need because you're looking at your model and the model says, well, I need this amount of RUP. Well, if you're really amino acid balancing, you don't need as much probably as what your model is saying. So take some RUP out of the diet. See what happens. If there's no negative response, okay, the cows are right, the model is wrong. RUP is more costly, obviously, than RDP. So to summarize, accept the basic aspects of protein and amino acid nutrition. Let go of balancing for crude protein, instead focus on balancing for RDP and the most limiting amino acid. Make good use of your cows. Use cow feedback to correct for model prediction errors insofar as you can. By all means, use reliable estimates of amino acid bioavailability for rumen-protected amino acid supplements. You're going to be really disappointed if you use some of these products and you're not using the right estimates. And use ration formulation programs with optimization capability. Hopefully your program gives you that opportunity. If not, encourage your software provider to include a more robust optimizer in your model that will give model compliant solutions that's fast and easy to use. You and your cows deserve that. With that, that's all I have to say. Thank you for your time and attention.